So there's two types of cells that are in eukaryotic organisms. There's somatic cells, which are known as diploid cells. They have two copies of chromosomes, one inherited from each parent. The somatic cells of the body, they're called body cells. They're going to undergo mitosis. We'll talk about another process in the future called meiosis, and that's when gametes are actually going to undergo meiosis and copy their chromosomes, but more on that later. Don't worry about the slide too much. Okay, here's the overall process of mitosis. You can see the first picture on the top left is G2 interphase. You can see that the chromosomes the DNA is still in chromatin form, but it's been duplicated at this point. Then we move into prophase, where you have the chromosomes condensing. You can start to see the chromosomes instead of just chromatin form. And then in prometaphase, you can see the nucleus starts to break down. The kinetochore microtubules, or the spindle fibers, are attaching to chromosomes, and the chromosomes are starting to move. During metaphase, the chromosomes are lined up along the metaphase plate. During anaphase, cohesin proteins holding the centromeres of chromosomes together start to break down by separase enzyme, and the dynein and kinesin motor molecules are going to help push the chromosomes apart and push the cell further apart. And then in telophase and cytokinesis, in telophase you see the nuclear envelope starting to reform, the chromosomes are going to relax into chromatin form, and cytokinesis is going to begin when the cell splits. Cytokinesis in animal cells is a little bit different than in plant cells. In cytokinesis in animal cells, because there's no cell wall, all that has to happen to split the two cells is that actin filaments are going to put together this thing called a contractile ring, and it's going to contract the plasma membrane between the two new daughter cells, and it's going to cause what is called a cleavage furrow. And that cleavage furrow is just going to get smaller and smaller and smaller and smaller as the actin filaments contract, and then you're going to have two new daughter cells once the process is complete. However, in plants, because there is a cell wall, you can't have a cleavage furrow because that cell wall is so strong and it doesn't just like fold in like a plasma membrane. So what happens is that there's a cell plate formed, and the cell plate is formed by vesicles carrying cell wall components in between the two new daughter nuclei. And this is where cytokinesis occurs, in the middle of the cell. And as you can see in that picture down there, the vesicles come together, they fuse, bringing together the cellulose molecules necessary to create a cell wall, and they're going to form that cell plate. And that cell plate is just going to extend in length until it's separated the two new cells, and you're going to get two new daughter cells from this process. And you can actually see that this is an actual picture of cytokinesis occurring in the plant cells, and you can see the vesicles coming together in the middle of that cell that's dividing, and the two new nuclei on either side. So scientists have been researching the process of cell division and its connection to ancestral organisms like prokaryotes. And they're trying to figure out how is binary fission, which occurs in prokaryotes and bacterial cells, how is that connected to what occurs in eukaryotes now? How did it come that these chromosomes that are in the eukaryotic cells can actually do this whole mechanism of mitosis? Well, we believe that the origins of mitosis came from the reproductive stages of binary fission. And binary fission is basically just when a bacterial cell duplicates its chromosomes and it splits in half. Instead of prokaryotes having mitotic spindles, because they only have one chromosome, so only one chromosome needs to get to either side of the cell for the new bacterial cell to be created, they have those origin of replication. And you can see in the highlighted green on the DNA, those origins of replication actually separate themselves, and they help separate the single circular bacterial chromosome because the origin of replication attaches to the either end of the dividing cell. So you end up with this attachment of the origin on either side of the cell, and it separates those single circular chromosomes. Now, over time, they've seen that there are some organisms that are single-celled, that are eukaryotic, called dinoflagellates and diatoms and yeast, and they actually have a nucleus, but they sort of follow the same sort of mechanism as the bacterial chromosome does. So their spindle fibers actually attach the chromosomes through the nucleus to either side of the outside cell, which is sort of an intermediate version of what bacterial cells do versus what we do in eukaryotic cells. And then the same thing happening in the diatoms in the yeast. The spindle fibers are attaching to either side of the nucleus, somewhat kind of like what happens in the bacterial chromosome with the origin of replication attaching to the cell membrane. And then the entire nucleus actually divides in the diatoms in the yeast. Now, in the most recent, in evolutionary terms, the most recent eukaryotes are actually going to lose the nucleus altogether and use the spindle fibers and the centrioles to pull the chromosomes apart. But you can see those protists kind of show us that there is an intermediate step between full-on binary fission that happens in prokaryotes versus mitosis, which actually happens in eukaryotes. So it's kind of interesting to see how this process of mitosis has evolved over time. Okay, I know this is very long. Hopefully you paused it to take some time, and I hope this was helpful.